Hello, everyone. Welcome to Foresight's Neurotech group. How we run these meetings is we'll sit for a presentation, and then afterwards we'll convene for a Q&A. But we're really excited today to have Bruce Hope, Mark Wells, and Greg Sutton here with us to talk about Nanofids. And we're really excited to have them here. This meeting has been a long time in the works. So with that, you can start the presentation.
All right. Actually, before I get going too far, I don't want to forget. I want to thank you, Lydia, and Foresight in general for having us here. And I also didn't want to forget to thank you for that, inviting us to that workshop, the Foresight Neurotech workshop in October. That was fantastic. I recommend it to anyone. And we met a lot of people we needed to meet. So they're a great bunch. It was fun. With that said, yeah, we've been at this for a while. And so there's Greg is here, thankfully, and Mark is there. And I don't want to leave out Oscar Vasquez Mena and Dugu Kuzum at UCSD, who have been major players in this project. But the main thing we've been trying to do is trying to make the promise of brain-computer interfaces, BCI, real. How would we make that full promise real? And so we've been working at this from first principles basis in the beginning. How do we push this technology along? I mean, like a lot of people, like a lot of people, there we go. We wanted to help people and use this BCI in a number of ways. There's, the obvious ones that people often refer to, like spinal disorders. If we could help people restore high resolution control of limb movements like this, it would be tremendous. And we already know BCI has been working on this kind of level of resolution for a while, but we want to do it with even more high resolution. But that's not where we want to stop. We want to keep going farther and, deal with more complex problems that BCI can help us with. And one, just for example, is like restore high resolution control of speech. I always think of what even Stephen Hawking was able to speak to us directly back when he was alive, and to help all people with these kinds of problems. And then even more complex kinds of things like high resolution control of all remote devices via thought. And the applications are endless, you can imagine. That's not difficult to figure. But I don't think BCI right now, the technology has really realized this promise quite yet. And someday, this is what we're really hoping, that we can provide that promise that BCI has been talking about forever. And eventually, provide high resolution integration, between AI and computer integration for whatever needs it happens to be. And the high resolution information about all the other kind of thoughts you can have, and about high resolution information about your life. All sorts of things, not just the more crude control of machines. And over time, we've had this thing like, what if you could read the full language of thought, not just fragments like are currently happening, but the whole complex symphony of abstract ideas, memories, emotions, and intentions that define what it means to be human. And we've been working on how do you make this happen? And there we go. The thing is about BCI is, as it is now, and it's some amazing people are doing some amazing work on BCI as it is now, but it requires many more magnitudes of bandwidth, like of getting neural activity out of the brain, than current technologies provide. So, the big problem, as we can all recognize, is that it's certainly not analyzing the data from the brain. The ability to analyze large sets of data, particularly with the huge advances in AI analysis, hardly makes this the bottleneck. And the real problem is, I think we're all extremely aware, is how do you get enough data out of the brain? This is the real bottleneck. And, electrode technology, for the most part, records hundreds, at most, maybe 1,000 neurons, like individual neurons. And that's key, as I'll get to, at a time. And that's a great start. I think they've done amazing work. But the fact is, you've got 30 billion neurons in the cortex. And, 100s, that's a great start, but we need to get to 30 billion. How do you do that? So that's been our goal, is to remove that bottleneck, developing a system by building a novel technology capable of recording millions to billions of neurons in human cortex with single neuron resolution for maximum brain computer integration. We've been working on this for some time.
Yeah, Bruce the reason this all got started is because Bruce had been watching the electrode method of, jamming hundreds of electrodes into the brain and seeing it not really working, and all the encumbrance of that. And so when the Galilean technology launch was based on the idea that the miniaturization of electronics had now finally reached a stage, even after us talking about this for a decade, it finally reached a stage where, we got together and thought, Yeah, I think in the next few years, we're going to have enough miniaturization to get, and the technology, the quantum dots and the graphene and things that are required to all come together to make these small 1 micron devices that could be inserted into the neurons. So, and luckily, Bruce is in a lab where we could do all those experiments to check how we could insert those in to get the maximum number out of those 30 billion neurons, to take up nanofids. And, there's, like all parts of this project, that went through a lot of different ideas about how to get them in there, how to keep them in there, how to stay in there, how to get them one-on-one to go into each neuron. And it took us a whole bunch of different ideas, which Bruce will probably go through some of them. But yeah, not only that, but, the thing about this project is, by working with the labs, we've not only had to discover how to put the nanofids in the brain by doing these microparticle experiments, but we had, each part of the whole thing, the calcium activation of the neurons inside the devices inside the neuron, and the many different parts. Our first ideas were mostly wrong. And we've had to go trial and error and try different things with all the labs running, Bruce's and the ones out here. And I think we finally have a solution that goes end to end, and all the things we've had to solve. There was about 10 or 12 big problems at the beginning, have all been solved. So I thought I'd mention that kind of this isn't just a concept, but really this is something where we've gone through many, a decade of trial and error with the labs, working out each step and trying it by making sure that each step works. And now finally, we did build some devices. Again, let Bruce talk about where we're going with the devices. So.
Oh, thanks, Mark. Appreciate that. Yeah, I don't want to give short shrift to the electrode folks. That is tough stuff, and the analysis, and they've made amazing advances. I don't want to give short shrift to them.
Yes.
So we've been developing a technology. So how do you get beyond what the current bottlenecks are? Well, so this is our system. I'll just describe it in some detail. I'll spend a bunch of time on this particular slide. And what I'm saying here, though, is that our device, we call them nanofids. It's our sensor. And these are about 1 micron size, and neurons will absorb these into them. We've already shown in my lab that neurons will absorb 1 micron particles just fine. They gulp things from the surrounding extracellular space all the time. This is what they do. This particular device, our sensor, is powered by a photovoltaic panel, and that's we're powered by light coming from the optical rays on the surface of the cortex. And this wasn't our first powering method. We have to admit, one of the iterations that we had before was bioelectric catalysis, which is basically living off the lay of the land, where enzymes on the surface of this device would actually catalyze the chemical energy from the inside of the cell and turn it into electrical energy. It wasn't enough for our purposes, wasn't a robust method. We also tried ultrasound for a while, but that also was not a workable plan at the 1 micron level. So photovoltaic panels, this is where Oscar Mini Vasquez was very fortunate for us to meet with him at UCSD. And he's been working on this project, making quantum dot photovoltaic panels, much like solar panels, except they're getting their photons from this optical array on the cortical surface. They power an LED, and this LED has different combinations of quantum dots inside them. And I imagine, some of and most people don't, but if you change the size of a quantum dot, it will emit a different photons of different frequencies or different wavelengths. But the point is, if you mix different combinations of these quantum dots in the LED, they will emit different combinations, and almost like emitting a barcode. Once they're activated, that can be picked up by the optical fiber array, saying, I am neuron number 44,304, that kind of thing. And then you need something that detects the activity. And this is where the calcium sensor comes in. And this is basically just stop me if you don't want me to get into this, but it's like a FET switch, a field effect transient. And it has molecules on it that bind calcium. Now, when calcium reaches the surface here, it increases the charge on the surface and allows current to flow through this circuit. And that's what allows the photovoltaic panel to activate the LED. I'll get to the logic layer later. But the point is, in a non-active neuron, this nanofid's not doing anything. But in active neurons in this cartoon, calcium rushes into the cell when it's activated. For those who don't do this kind of work, when neurons strongly activated, calcium rushes into the cell and raises the concentration inside about 10 to 100 fold. And this is the kind of stuff that already people are using with proteins in the GCamp cell imaging field. But we developed an electronic version of that has barcodes. Now, once that barcode signal goes to the optical array, these are just collectors. They're collecting photons. It doesn't matter where the photon's coming from. One thing I left out here is that brain tissue, like most tissue, is relatively, shall we say, it doesn't absorb photons. Photons will bounce around. They're heavily scattered. And that's why when photons from this particular nanofid, they bounce all over the place. But as long as they're collected by the optical fiber array within that 10 nanoseconds, then it will identify that particular neuron has been activated. And these fibers will be collecting these photons, but you have to separate them in a rapid way. There's going to be a lot of data coming out of this device, a whole bunch of devices. And so this is where Greg's has this system, developed this system using 30 different fiber Bragg gratings in series to do rapid optical, basically signal processing to identify these neurons very rapidly. And somebody's trying to call me, I'm going to decline. So very rapid signal processing so that we can turn analog to digital signals without hardly any computer processing ability. Did you want to say anything about that, Greg?
No, I think that was good.
All right, cool. And but there's basically the idea is that these you can inject just basal level, starting position. It should be known that in neurosurgeon world that a 3 centimeter hole is about like the size of what they call a keyhole skull opening. And I was surprised as anyone that's considered minimally invasive, but apparently they do this kind of work all the time. So that's not a problem. But there's 70 million neurons accessible inside this minimally invasive keyhole skull that would be possible for us to inject nanofids and absorbed into the neurons and have optical fibers over that. The specifics of where the optical fibers leave the brain or the skull and go under the scalp, that's another thing entirely. But basically all this information, optical information, is rapidly processed at literally the speed of light so that it can be analyzed or control a variety of machines to interact. But I want you to notice these things are small, 1 micron, much, much smaller than the neurons they're recording from. This is a key thing I'll get to later on. Signal processing is very rapid. Also, the nanofids are inside the cell, not exposed to the immune system or otherwise physically moving relative to the neurons. So there we go. So I want to compare it now to the other technologies that are out there. And I definitely want to start by saying I appreciate the immense amount of knowledge and work that's gone on before. It's fantastic. But in this comparison here, I just want to back up just for a moment to talk about basically how information is encoded by neuronal ensembles for the people who don't do this kind of work. And I'm just drawing a cartoon to make it simple. But basically, in the cortex and other brain areas for that matter, a thought or behavior, or it can be encoded by a specific pattern of activated neurons. And if you go to a different pattern encodes a different behavior thought, and you could even encode memories if there's physical changes within these particular ensembles. And they coexist within the same brain area, and not just coexist, but they can overlap as well. I mean, there's many overlapping patterns here. Any one neuron to store all of these thousands and millions of memories in the cortex, you're going to have a lot of overlap. Any one neuron is probably encoding thousands, if not many thousands of neurons, memories, I should say. This being the case, you cannot say that a neuron is encoding a particular behavior, thought, or memory. As I always like to say, the message is in the pattern, not an individual neuron, but the pattern. And I'm getting to the point of why this is useful for comparing the different technologies. And the best way I think of thinking about this is this: we're trying to find the QR code of the brain. There are specific patterns of, pixels here. Think of them as neurons. Pixels could be used in multiple codes, but it's still the pattern that gives you that particular signal, the hyperlink for your favorite restaurant versus some hospital. That's the way to think about it. And so we're building a system to read all these QR code patterns in real time, from first principles. And I always like to say, engineering needs to fit the brain, not the other way around. And using this ensemble-based information, how that encodes all this information, creates certain strict design constraints on the BCI that would be useful for brain AI integration. One is single neuron resolution is critical, because if you're looking at more than one neuron from a sensor, you're actually looking at a whole muddle of different signals, encoding various and different memories and different thoughts. The neuron right next to it can be encoding a completely different memory or thought or behavior. And due to this high overlap, as I mentioned, any one neuron's involved in thousands of different kinds of memories or behavior. Because of that, these patterns are very complex. And to distinguish them in a very meaningful way, to make you sure you disentangle memories and behaviors and thoughts when you're recording, you need to record millions of neurons to distinguish these patterns. So with that in mind, I'd like to do a comparison. So existing electrode technologies, they've been around for, a very long time. With Vernon Mountcastle back in 1957, and he was studying these kind of sensory recordings and motor recordings with electrodes for the first time back at Johns Hopkins. And he's obviously one of the most famous people in neuroscience. But this kind of technology existed for a very long time and doing recordings. But one of the most important problems was that when you stick an electrode in, you're either displacing or destroying neurons to get to the neuron that you're recording from. Part of that was solved using microelectromechanical technologies. And I took this picture from Jürgen Bijaki's review in Nature Neuroscience. The idea is that they would place multiple sensors, electrodes, on each of these shafts that they would put inside in the brain. And this solved some of the problem. So like being able to get multiple electrodes in without destroying too much of the tissue. And the next piece for comparison is one needs to know that there's about 100,000 pyramidal neurons, roughly, under each square millimeter of cortical surface. And when I'm talking about recording density, I'm talking about the number of neurons you can record from per millimeter squared out of that possible 100,000 neurons per millimeter squared. So when you look at some of the current PCI technologies, just take the most talked about one, Neuralink. If you do the calculations, how many electrodes per millimeter squared, it works out to roughly 7.2 electrodes per millimeter squared, which is a 0.007% recording density. You could probably disentangle multiple neurons that are being recorded at a given neuron. So maybe let's give it 0.02% recording density. It's good, but there are also many other companies, very talented companies out there. Now, NeuroPixels is more of an academic group, but their recording densities, at least in the lab, can get as high as, say, 0.5% recording density using the same kind of calculations. And then one of the most established companies, BlackRock Tech, they get about 0.025%. And then Peridrone, which I guess came as an offshoot of Neuralink, they're getting up higher, around 0.1% recording density when you use this particular calculation method, using what information I've got anyways. And this is good, and it's certainly solved a bunch of problems as we've seen, but it still runs into the problem of what is called the butcher rate. That number of neurons that are destroyed or displaced by the electrodes or the shafts that go into there relative to the number of recorded neurons. So you can imagine, even if your sensor is the same size as a neuron, then that means you're going to be displacing, if you're doing 100% coverage of the brain, you're going to be displacing all the neurons in the brain. It's not physically possible. And this is a major problem that has to be solved. So the butcher rate, in my calculations, makes it unlikely for electrode technology to get more than, let's just say, 1% under the best of conditions. And so that's great. I think that's not bad. But electrode technologies do have the single neuron technology that it needs. Its recording density is not bad, but I'm going to show you a technology that can push us farther. Scalability, the head stages do become a problem where you can't have too many on there. And then when you're recording from thousands to perhaps millions of neurons with electrodes, it would make it hard to do the signal processing. It's going to be a big problem. Then, of course, movement artifacts. Movement artifacts is when, see the electrode is kind of in a fixed position, but when the brain moves, a millimeter here, 2 millimeters there, you're going to lose that signal. This is a major problem when the person, or animals if you want, are moving around. And the other issue, which is actually getting better and better nowadays, is the lifetime of the recorder, or actually the sensor, I should say. These electrodes are all outside of the neuron, and they're exposed to the brain's immune system, and they are also physically moving relative to the neuron. All of these things activate the inflammatory responses of the brain. And over time, will encapsulate them and minimize the electrode's ability to sense neuronal activity. So with all of this in mind, we were incorporating all of these ideas into what kind of technology would make to get us past this. And that's why we came up with the advantages of our nanofid system. One, these nanofids are extremely small. Their relative ratio in size to the neurons, let's just say our average 17 micron diameter pyramidal neuron, it's basically taking up 0.1% of the volume. So its displacement of neurons will be minimal compared to things like electrodes. It solves part of that problem. And we don't want wires connecting nanofids or whatever sensor we got inside the cell to the outside of the brain, because that would also take volume. And that's why we have to have a remote detection system like the optical fiber array. These are some very basic first principles that have to be involved in any technology that solves the problems that we currently have with the electrodes. The other thing is these, when I've injected, 1 micron particles and do electron microscopy, the particles are not associated with degradation organelles, and these things are inside the cell, so they're not exposed to the immune system. So it solves that problem as well. And movement in this system, you could have the neurons moving relative to the optical array, millimeter here, millimeter there, and that's going to increase or decrease the strength of the signal, but it's not going to change the barcode identification system. A lot of people might say, well, doesn't the intense light scattering in brain tissue cause a problem? It actually helps us. I know it really is a huge thing in calcium imaging, a big problem for them. But for us, we are not counting on what position, like spatial position, the active neuron is in relationship to the optical fiber array. All that matters is that specific barcode is received and collected by those optical fiber arrays. Then we can identify the frequencies and wavelengths of that barcode. And that's how we do it. So the intense light scattering is actually to our advantage, because might say, oh, this nanofid's, turn this way relative to the optical fiber array, and it would absorb photons different from the optical fiber array for energy, or emit them. That might be a problem. But the fact is, scattering is so intense that it really doesn't matter about the orientation. And so that's our friend. And one thing I should have said before is a paper I published some time ago, that neurons naturally absorb these 1 micron particles. And as far as we can tell, with no apparent damage. But we have to do a lot more research on that. Right now, I am currently optimizing the distribution of these particles within the brain, particularly the prefrontal cortex in animals. So in many ways, even just on first principles, this is a technology that would be capable of doing all this high resolution integration, which was always been our goal. And so that's what we've been working on. But I don't want to act like this is going to take some time to get from one place to the other. We've done a lot of things, as Mark has mentioned. We've built a variety of components at the right sizes. We've looked at the absorption of particles by neurons and all this that's required for it. But I think anybody seeing this project would say, yeah, you don't build that right from the start. So I want to tell everyone what we've developed a first stage project that will allow us to get there much faster. And I have to say, this is one of those moments where I was at Foresight's Neurotech workshop in San Francisco in October, and there was a guy there who actually, in a conversation with him, got me to think more accurately about this. And so Greg, Mark, and I have started to design a more simpler version to start with. And I'll show you this in a second. But we needed something that was simpler to start with. So first of all,
we need something quick. We talked to some funding groups, and our original project for the 1 micron is kind of a 5 to 10 year project. And to get to pre-FDA stage, because of all the machinery you'd have to build for the micro miniaturization. And this project here will allow us to do at least half of what we want to do, and still get a higher resolution that we're targeting. But we've already built 20 micron devices, so not with all the pieces in it, but we built ones with the quantum dots and with sensors on them, and so we know we can build 20 micron devices in the clean rooms we have available to us, because we already have. So we're going to go ahead with this, and then we'll be able to we'll use some of the aspects of this in the final nanofid design. So we're kind of excited that this is a 3 to 5 year project, possibly 2 to 3 year project. Things really fall together quickly. So we're excited about being able to show some of what we could do. It won't be 30 billion, but it could be neurons getting recorded. So that's a big more the state of the art is right now. Sorry. Yeah.
No, I appreciate that, Mark. Thanks. Yeah. Often I forget to add things, but yeah, we were excited about doing this stripped down version because this is something we could do now. So we're actually, putting together the plan to build this right now. And but this first stage version, it's 20 microns that makes the, manufacture a lot easier. But two, we're not doing it's not going to be inside the cell to start. We can do extracellular recordings, and we're going to use multi-sensor. Now, this sounds a lot like, I'm sure to some people who follow this, more like neural dust from like the related things like from the Nurmico and the MIRB's labs. But it's basically the same system, but the sensor has changed, to a voltage sensor. It doesn't have that calcium binding molecules on the field effect transistor, and they will be implanted using some techniques I'm doing in my lab extracellularly. So, and then they will record the equivalent of local field potentials, not unlike what electrodes are doing, to be honest. But beauty of this is, number one, sets up the prototype for us shrinking down the devices. Number two, these devices, like neural dust for that matter, will move with the tissue and not have those problems. It's optical processing, as in the full system, and which make it very rapid signal processing, rapid identification of the signals. And not only that, it's something we can do right away. And that's something we're excited about because that makes it a much more project that we can do right away. And it would produce recording densities of at least 1% recording density if the calculations are correct. And we can certainly tweak this system. We'd be even happy with this system. But the second stage from there, that our plan is, our strategy, is doing certain changes. Let's say, there we go. A second stage product. Once we've established that, make some not minor changes, but some substantial changes, like 1 micron size intracellular version instead, which is one change from 20 to 1, extracellular to intracellular version. And we're going to have it somewhat more sparse. And the recording density will be 1% again, but at least we get absolute single neuron resolution. The other thing we will be adding the calcium sensor to this system. And at this point, we're almost there for the full system. The only thing that we're going to end up adding is what we call input coding, by having these thin layer notch filters that we'd be getting from Alexa. In this way, we have distinct sets of these particular nanofids that we can have both an input and output coding happening to allow us up to about 2,740,500 different unique combinations that allow us to identify the 1.9 million neurons under each 7-fiber array of optical fibers, which I can get to. And this is the system and the strategy that we're using to get to this final full system. This is our vision for all of this. And this is the comparisons, basically, like 20 microns to 1 microns, extracellular to intracellular. In the beginning, it will be LFPs, local field potentials, to single neuron, voltage sensor to calcium sensor. And the number of codes that we can do in the beginning will be, giving us 1% reading. But the Alexa filter, once we develop all the other things, that will be the final step that will give us the full 100% coverage. And then 100% coverage. And the next slide is, because people keep asking us, and I keep throwing it in there, people keep asking, how do you close the loop using this system? Well, I'm just going to throw it out there. And using a similar concepts, we won't be just reading the output. This is way down the road. I don't want to act like we're doing this now. But writing input to neurons in brain. How do you do that?
Yeah, so by closing the loop, you mean it's like, communicate back to the neurons to activate single or patterns of neurons back to the brain, which of course the military is already told us they're very interested in, and for soldiers or for animals. Yeah, so, and we've done some project with DARPA already, but anyways, being able to go both directions, not only read the brain, but write the brain is an outcome they'd really like. So go ahead, Bruce.
Right. No, that's great. And, to do this, we could use a similar concept, and it could be the same nanofid device, or it could be a separate activation device. And it focuses on, this is just one possible method of many, but there's the exon initiation, exponential, you even say, initiation segment. This is one of the lowest threshold for activation parts of a neuron. And in this area, if it gets depolarized, it will activate, the entire neuron, all the way down the axon, but also the rest of the cell body. And the idea is that if you had a device that was anchored to, say, near sodium channels, for instance, in this area, you can have one that uses these electroactivated polymers to move an activator peptide, of which could be these scorpions and spider toxins that are known to bind to these sodium channels, and therefore activate the sodium channel, and in such a way that, you turn it on and turn it off. Obviously, how the heck are you going to identify the specific device that will activate the specific neuron of interest? I'm not saying, I'm just throwing it out there. Once you've got an electronic platform out there, there is simply a logic layer for serial inputs selection. And that could be a 16-bit code to identify that 27,405 different possible devices in a given area, and multiplied by the Alexa filters that give us a different additional 100 times input code. So this would be serial.
So we have to start a spider and scorpion farm for this?
Yeah. I like spiders. I like scorpions.
When you first said spiders and scorpions, I was thinking you don't really mean spiders and scorpions, but I thought, no, he does mean spiders and scorpions. Venom. Yeah. Okay.
Yeah, but evolution and biology has provided us with a lot of fun tools. And I like to sort of delve in that strange space between machine, engineering and biology. That's my favorite place where the interactions happen. And like I'm saying, there are a number of different ways of doing it, but this is kind of system. You need something to identify the specific system. You use that input power as to serially select the activator peptides to activate a system in a neuron. But who knows when that will be? That's down the road. So I'm going to let Mark take this. Mark, you tell him.
All right. Yeah. To close Bruce's fantastic presentation, we just want to tell everybody that we've got, we've received NIH and DARPA funding to help us get here, and we had support from UCSD, UCLA, UC Santa Barbara, and ongoing long-time support from NIH, and continuing support, and all those labs continuing to help us work on that on every day, working on refining all the solutions. So, and we want to. The big challenge for us is to fund building the machineries for the devices in our own facility. We'll use the labs as long as we can, but there's some things that are going to be very customized. They probably don't want us cutting into their machinery to make our own processes at UCSD and UCLA. So we need funding to do that. So we're working on raising our A round of funding. And, we have some ARPA-H, and we're still working with them. But, their comments were, we're audaciously innovative, revolutionary technology. It bypasses all issues of other BCI, and they're very enthusiastic. We have 4 patents. Two have been granted to our process right now on this revolutionary approach. And so we hope that we can eventually get funding help on this project to take it to the FDA stage.
Yeah. And so do you not care about this slide, Mark?
Oh, it's good. Yeah. So yeah, thank you. Oh yeah, there's the next slide. And so that's basically, we're raising $15 million on a $50 million valuation Series A raise. And we want to get involved with venture capital or investors who are excited about connecting AI to the brain and having a symbiosis type of connection that we can eventually share memories and thoughts directly with AI, and the other way around. Thank you very much, Lydia, and everybody at Foresight for supporting us in this.
Yeah, appreciate it. Should I stop sharing there, Lydia? Or
maybe we can leave it up, because now we'll go into our Q&A section, and maybe you'll want to reference some slides.
Absolutely. I have a whole lot of slides, some of the very science nerdy stuff if they want it. And I'm not saying I'm going to have the answers to everything. I'm not saying there's so many smart people out there. I'm sure I'm going to be befuddled by some of them.
Yeah, I saw a whole bunch of questions come in. Are you able to read
there
those,
are so many questions. So right to it.
Yeah.
I think maybe Bryce, would you like to go?
Sure. As I mentioned, this seems like a really interesting approach. Very much excited by the prospect of the sheer information density it sounds like you can get out. My background, beyond the technical side, I'm working on an MD at Stanford. And so I immediately start thinking about the potential anatomical, physiological problems that can arise. Neural tissue, incredibly delicate, really inferior so when it comes to developing BCIs. And so, as I mentioned in my comment, there's a couple of immediate concerns that come to mind. First one is, you said that it's not immunogenic because it's intracellular, but you still are going to have the brain's immune system exposed to the nanofids when you install it. Neurons turn over time, which means there'll be continued exposure. And so do you guys have a sense, have you been able to do any experiments looking at how that reactivity progresses? Do you get reactivity? And the second question is, once they're in the neurons themselves, if they're micron scale, so reasonably small, but if they induce any structural impact, or especially if they induce any like aggregation processes, that could readily lead to dementia in the long run. So I wonder how you guys are addressing those kinds of concerns.
I can tell you that we're going after every single one of those questions, but right now we're at the stage of just microparticle distribution. I can tell you that from our own information that we have not seen like clustering or any observations of any differences using electron microscopy around the silicon microparticles that we've used as our model systems. We are doing experiments right now, actually been doing quite a bit of them lately, but just using these fluorescent microparticles, and also we are examining using IBA and GFAP antibodies to identify, various easy inflammatory markers. But this is just beginning right now.
One thing to mention, though, is that the nanofids are coated with silicon carbide, except for obviously the area where they're activating on either the voltage or calcium reaction. So the silicon carbide is very inert, and we think that's a key thing for making sure that the interaction doesn't happen. Did I say that right, Bruce?
Yeah. But silicon carbide, I know, has definitely been the thing in electrode technologies as well. And is that this inflammatory response that Bryce brought up is very key. And we just haven't got to that part of it in our particular process yet.
Got it. Thanks.
Great. Next, there are 2 questions in the chat I want to highlight. The first one is from Benjamin. If you'd like to ask it, or I can also read it, your question.
Yeah, sure, I can ask it. Thank you for the talk. That was really exciting. I was just wondering, what happens to the nanofids if a neuron happens to die?
Yeah, sometimes I have to just fess up and say, after all the time I've been thinking about this, that has not really, to be honest, occurred to me. I didn't even think about that as a question.
I never thought of that before either.
Yeah.
That's crazy.
I guess that's what we get. It's absolutely true. 100% true, because neurons do past the age of 18. A lot of neurons die every single day. Yeah, I'll be honest, I don't know the answer to that, but I do know this, that when we've been injecting silicon microparticles, we haven't noticed any like IBA or GFAP response for inflammatory response. We're also the amount of, perhaps materials that
I have to jump on the call for 1 second. With video.
Okay.
Do you mind if I?
No, please, yeah. Oh, Philip. Oh my gosh. Okay, hi. But is the amount of possible problems, like quantum dots are made of things that are not too kind to the neurons, but they're all encapsulated within, silicon, et cetera. But secondly, they are also, the amount of it is so diminishingly small that it's well below what people consider toxic in the brain. But that said, it's all going to have to be tested. But no, that's a great question. There's going to be a lot of dying neurons, even in normal processes.
Great. Next one in the chat, then we'll move to the raised hands. We have a question from Gert.
Yes, and I think it was already addressed.
Okay.
But yes, so if you have a micron-sized nanofid, how does it enter the membrane without disrupting the membrane?
Well, first of all, the precise type of endocytosis, I can't tell you, but I don't know. But the neurons, like most cells, are constantly drinking, you could say, from the extracellular space. This is what all cells do. And people used to think that neurons couldn't do anything bigger than maybe 0.5 micron, but we showed that was not the case. It's not just the macrophages and microglia that are doing that, but I would like to know what specific form, probably of endocytosis that mechanism is, but it's a natural mechanism. It's nothing that I don't think we forced any unique action by the neuron to get them to go inside. I believe that this is just a natural endocyte process. Somebody determined to get a hold of me so that's the end. From what we've seen, again, once it's endocytosed by electron microscopy, it seems to be in the cytoplasm just fine. It's not around lysosomes or any other kind of, degraded some. So that's all I can tell you right now at this point.
Yeah,
from what I see.
Because molecular machinery for endocytosis, and it needs to be very specific for this one. Philip has many other questions. I think that.
Yeah. I remember Philip was the one who got me thinking about that extracellular stage 1 system.
I guess that means it's my turn then. I think I had my mic on earlier accidentally, so apologies for that. Good to see you again, Bruce.
Good to see you.
Just a couple of questions. I think the question of removal is an interesting one, but that may be something that eventually will be accepted. But when you think about a micron scale device in the parenchymal space, it doesn't really feel like that's going to diffuse very far. So how do you expect to deliver to a volume? Have you done any demos of it?
No, but this is exactly what my staff scientist is doing with me right now. And we're looking at different ways. What we're doing is we're using these microparticles, 1 micron size microparticles from Micromod, German company, and they're fluorescently labeled. In our case, the optimal one was Psi5 in the far red. And currently we're tested different surface types for hopefully the ones that do not interact with the extracellular proteins or extracellular matrix in particular. 2 other things we're doing is, there's a couple of simple ones basically. And mannitol is, 20-25% will expand the extracellular matrix. And this is stuff that is ongoing basically, and they do this for clinical reasons too, to expand the extracellular matrix to enhance that diffusion. That's one thing. I'm hoping we don't need to go, after those 2 things I've mentioned, I'm hoping we don't need to use these methods, but I think it's a very temporary thing, is hyaluronidase, using what some people call it breaks down hyaluronic acid in the extracellular matrix and allow more easy access to particles. People are using this for studying nanoparticle distribution. And hyaluronic acid is basically created quite rapidly to basically close up after such a distribution. So we're using the 3 different things is what we're examining to enhance distribution. Because yes, they absorb. I know the neurons absorb these particles, but to enhance their even distribution. That is like job number 1 for me, is getting even distribution of these things.
I'll let others jump in.
Yeah. We'll call them the Zoom hands. Marcus, would you like to ask your question?
The Zoom hand.
Yes. Well, I got lots of questions. I'm not sure whether we have time for all of them, but I was involved in the CAN-DO project, optogenetic stimulation for epilepsy patients. And for optogenetics, one question is always, you have LEDs and they generate heat, are they heating up the tissue? So in your case, you basically have this big optic fiber bundle for powering the system 100% of the time. How does it change the temperature of the tissue?
Yep. We're not there yet in our method, okay, but we did the calculations based off of work from who've done it. It's in the supplemental section from one of the Stanford folks, one of the first to do optogenetics with Desiree not Lefkowitz, but I'll remember his name later on. But we used his, and also some work from people doing 2-photon microscopy, because overall there is not data out there using optical fibers with near infrared. Usually it's invisible wavelengths. And visible wavelengths are absorbed by brain tissue about 30 to 50 times more than near infrared, which is the range we're operating in, between 700 and 1100 nanometers. But based on the work that others have done, looking at how neuron activity is altered or what can actually damage the cells, it's about, 600 nits per square micron, basically is the max that we can do. Well, not the max, but it was safely shown by those groups. So that's as far as we need to go. And we've done all the other, simulations for how light scatters through the brain. And we don't need any more than 600 nanowatts per square micron. And because that will actually activate our devices just fine based on our calculations of how much energy, not our calculations, but actually experiments for how much energy we need for these devices. So a lot of these measurements about heating up the brain, a lot of people they're doing it in spaces or doing it in anesthetized animals where blood flow is very low. And it's blood flow that takes away that heat. So you really need to do those measurements, not like above the skull or, in certain meninges, but basically in space where there's high blood flow, like the brain. And so we've done those calculations, and I only have it in text format, so I don't have a pretty picture or cartoon for you, but we're well within the range of heat generation inside, and it will be actually very low for using near infrared.
Yeah. Okay. Just very quickly, sorry. So you have those neurons that are sending out the barcodes. So if you have 2 neurons nearby activated at the same time, so they send barcodes at the same time, do you get any interference? Or
we definitely could get such interference, but okay, I am going to have to stop for a second and then move along on this here. So the way to solve that was remember that logic layer, maybe at the beginning of the talk when I was talking about the design? Here, I'll have to share this first, but here, I'm just going to bring it up. Sorry, I didn't mean to have to go in and out like that, but basically, this is a problem that, continue to share. Thank you very much. When there is a calcium signal, it's a fast rise, as a lot of you people already know from calcium imaging and It's within about 5 milliseconds. I'm just rounding things here. But there is a very long, basically decrease, the decay time. It can last half a second, 1 second. And there's going to be like at least 190,000 of these things, like 10% of the neurons just say, active within any given second under the 7-fiber array, which I can get into. But let's just say they're all activated within about 25 milliseconds over 1 second. We won't be able to distinguish more than 2 to 3 of these co-emissions from nanofids next to each other, as you brought up. Our solution to this, and it was actually from the electrical engineering world, turned out to be a lot simpler than that, is just basically catch the rising edge when there's a certain threshold rising calcium concentration. Then you only send a 10 nanosecond burst of that barcode that will identify it. So even if 190,000 neurons are co-activated within 25 milliseconds for an average spread of about 132 to 1320 nanoseconds, a 10 nanosecond emission have very few overlapping emissions under these circumstances. And this is with a lot of neurons, 10% of them firing within 25 milliseconds. So that's the system. That's the strategy we're doing. And
we basically can multiplex electronics much quicker than systems can turn a neuron on and off. So we have a chance to, there's the timing diagram. So you could do many, it's like a keypad. I always think of you strobing sets of neurons at once and going through them all while they're all just like slowly changing. So you have many chances to electronically strobe them because electronics and light are so fast compared to the neurons that are going on and off. So that's the hack for, you know, to get a bunch of them literally side by side, which may or may not be strobed at the same time, but probably won't be.
Yeah. So okay, thanks.
Yeah, it's with the input coding, through the Alexa filters, and you can control when they're are capable of responding, and that's how we do that. And we even with the cooling aspects, we can even slow down the input power and record just fine as long as we have a pair of 2.5 millisecond pulses. We can play with that too.
Awesome. I know we're over time, and I want to be respectful of everyone's time here. Would you have time for one more question?
Yeah, of course. Love to do this. It's fun.
Great. Micah, I saw you had your hand up, but you put it down. Do you still have your question, or was it answered?
I still have questions, but I'll let other people go. There's other questions.
Okay. Well, then our last question will be from Jay.
Hey, Jay.
Hey, Bruce.
Hey.
So I'm a little bit confused in terms of what signal are you detecting from the neuron? What does it tell you about what the neuron is doing? And how do you interpret that over 100 billion signals, assuming they're distinguishable?
Yep. Well, assuming that everything works as claimed, they will be distinguishable, but we'll find that out. And the intracellular calcium, so the devices, at least with the microparticles that I've used as a model of those devices, they show up in the cytoplasm. They don't show up next to any particular organelle or next to the membrane particular.
And so I guess I sort of understand the calcium part.
You got that.
But I'm wondering, what is the dynamic range? Like what exactly are you learning from that neuron? The experiences of that neuron in a given thought or behavior, and how does that translate? How do you interpret that?
Yep. Brings up the whole second stage of this is that our biggest job is to just get data out of the brain. Okay. And then at that point in time, provide the meaning of those particular patterns, or you could even stretch the analogy I gave in the beginning of, the meaning of that particular QR code. That's going to have to be developing the software for that, of the brain, pairing up the behaviors, the thoughts, emotions that are ongoing with that particular pattern. And I have to say, when we're getting into that kind of analysis, we're getting a little far from my knowledge base, but this is where big data, AI analysis, and all of that are going to have to pair the QR codes with those thoughts and emotions. And I'm gathering you're going to be the person who could tell me how to do that better than me. So,
all right. Well, let me know when you're at that stage. I'm glad.
Perfect.
Just to help.
I'd be happy to.
Just one more follow-up. I know there's time is a problem, but do you have plans to try this on a simpler organism before you go to the human?
Oh, heavens, yes. I'm a rat guy, so we're definitely going to be doing this in rats long before then. And I do have lots of colleagues who work on primates as well, so we're definitely going to test that out long before. But I do ensemble work in rats.
I was actually even thinking of maybe Drosophila, if that's even feasible.
I have colleagues who do that. You work with them. It's a messy business, flies all over the place, but it's definitely a system we could do.
Great.
I think it would be very interesting. It's not just for research. I mean, not just for, curing everything too. There's very obvious research applications for understanding what all these large data activity sets can mean. We just haven't had large data sets. I mean, super large data sets from single neurons to work with. I mean, we just haven't gotten there. So that's going to be the fun part. So doing it with Drosophila or rats and all of that is going to be incredibly important.
And then you'll have to distinguish between neurons and glia.
Yeah, absolutely. From what we've seen, yes, some do go glia, no doubt. But also when we were doing the double labeling.
I mean, I would want to see them in glia as a neurobiologist, because I think the glia are underrepresented in our understanding of how the brain works.
I happen to agree with you very powerfully, especially since my son, Keenan Hope, he's doing his PhD, work with glia up at Case Western in Cleveland. So he's certainly, convincing me to look at glia a lot more strongly, because I know it's the relatively unknown, underappreciated cells in the brain that I think are part of this signaling. I think it'd be really important to know about.
Absolutely. I thank you.
Thank you, actually.
Yeah. What a great presentation. Thank you so much, Bruce, Mark, and Greg,
Lydia.
and for everyone for bringing your great questions.
Thank you.
Yeah.
Thank you.
Is there any
very much.
email that people can email you, maybe if they have outstanding questions or
Yes,
comments?
absolutely. And you can get me as well. But here, actually, right at the beginning here. Here's my email here, I'll blow it up a bit. But you can see my email here, bhope here, as well as Mark's and Greg's here.
Mark.
I can leave that up.
Yeah, it's just Mark at gallianatechnology.com, and we're the same with Greg, same idea. So yeah, feel free to reach out and ask any of us questions and getting involved with our business, the company, our vision of nanotechnology. We'd be really excited to work with anybody else.
Yeah, absolutely, man. Absolutely. And I guess the other way is you could just scan this with your phone and you'll get, it's gallianatechnology.com. It's not hard to find us there. So
Awesome. Thank you all. And we'll see you next month.